So you might be wondering, although I doubt it because you overlap with that today, maybe you were wondering why we're going on chapter 9 and then all the way to the end of chapter 28. So here's the rationale. This begins sort of our diversity unit. And one of the things that we're going to talk about is the diversity of microorganisms, but in that we're going to talk about two ways that microorganisms are utilized because of their diversity. So in chapter 9, a particular aspect of biotechnology, and biotechnology is a really big word that basically means, right, technology using some organism or something from an organism. So it's a vast topic. I mean, you can get a degree in biotechnology, you can work at a biotech company, right, you can do all sorts of things. So what we are going to do is look at a very small section of biotechnology where microbes are used for some medical purpose. So either to make things or because enzymes have been extracted from them or different things like that, right? So sort of the medical microbiology section of biotechnology. And then really chapter 28 is biotechnology. It's just typically industrial. So we're going to look at those, bless you, those two chapters together because both of them give us a glimpse into how microorganisms are used either medically, industrially, in food preparation, or different things like that. So that's why we're looking at them together. We are not going to look at everything in either of those chapters. You might have guessed that from the fact that we're looking at 2 chapters in one day. So we're just doing a real brief survey of these. So when we do brief surveys, keep that in mind when you're doing your studying, and don't go read every page in the textbook thinking that we're going to be studying everything, because we're not. Right? So chapter 9, basically we're going to be talking not about recombinant cells per se, but about recombinant DNA, which is the same thing as recombinant cell, right? A recombinant cell, remember, is that recipient cell who has received DNA from a donor. So they now have their DNA and also DNA from the donor cell, right? On Tuesday, next Tuesday, you're going to be needing to remember that for your exam when we talk about transformation and transduction and conjugation, right? But that cell is recombined because there's a recombination event in the DNA. And so in chapter 9, we kind of look more closely at what happens in that DNA molecule. But we're going to look at a very small section of biotechnology looking at recombinant cells, which themselves have recombinant DNA. So in chapter 8, it's sort of a more what you would call in vivo approach, just natural things that happen in nature, right? In chapter 9 and chapter 28, we're going to talk about in vitro, which means in glass, which means a lab application, right? So chapter 8 is kind of transformation naturally happens, conjugation naturally happens, transduction naturally happens. Chapter 9 is, and chapter 28 is, these natural phenomena, how can I use them to some particular end, right? So we're going to talk about using something called a vector to produce clones, right? And a vector is not like a mathematical vector. A vector means something that delivers DNA into a cell. For instance, a virus. In the process of transduction that you learn in chapter 8, you have a virus delivering DNA into a cell. So we're going to talk about that. We're going to talk about some ways that that DNA gets inside the cell and then can be seen there and can be used there, and this has wide-ranging applications. This process that we're going to talk about, because we're just going to talk about the process, can be used to produce human enzymes and other proteins, right? Human growth hormone, insulin, all sorts of fun things. It's also used to make vaccines. It's used for gene therapy. And it's used for disease identification. So these are just kind of some of the things that you could do with the tools that are in chapter 9. Because really chapter 9 is sort of a tool belt, right? You got a hammer, you got a screwdriver, you got your drill, right? What are you going to build with those things? And this is kind of all the things that you could build, right? What we're going to talk about is the tools that are in your tool belt. And then you can just think that those things can be used to all sorts of different aims, some of which we're going to talk about in chapter 28. Right? So that's sort of the area, right? The cloning of genes into cells and then expressing those genes, and that is the portion of recombinant DNA technology that we're going to look at, right? So you're going to get used to seeing this recombinant word, you're going to see R in front of DNA, you're going to see R in front of cells, and that's always going to say there's been some kind of new combination of DNA from what existed already and what has been added to the cell. So the basic process, and this is not the only process, but this is the only process we are going to talk about, right? So there's lots of them. But the way that we're primarily going to talk about DNA getting into a cell and becoming useful is through this schematic right here, right? You start with a bacterial cell, because bacteria are easy to grow, they grow quickly, and they're little, so we can grow them in small areas, right? So bacteria are a great choice for who's going to make your protein, right? Because people also make proteins, but people are big, they don't really like getting poked with needles, and you might get a disease on top of that, right? Because let's say insulin, for example, right? It used to be, if you were an insulin-dependent diabetic, that they would crush the pancreas, usually from a pig, and they would try to extract the insulin out of that, and that's what you would take therapeutically, right? And you're like, ooh, gross, but otherwise you would die. Today we don't do that, right? We don't extract insulin from pigs. We teach microorganisms how to make human insulin, and then we extract it from the microorganism, so it's a much cleaner process with much less risk of disease transmission. So we start with a bacterial cell, but we have to teach that bacterial cell what to make, right? We have to give it the recipe, and the recipe comes from you or from some other thing, right? So if we're talking about insulin, then we would say in the huge human genome, we have this gene of interest, and it's the insulin gene. We need to cut that recipe out of the human cookbook, and we need to insert that recipe into the E. coli cookbook. And we can do that by taking that recipe and putting it into a plasmid, right? Because remember, we talked about a process called transformation. And transformation is just when DNA in solution goes into a cell, right? And the great thing about that DNA, when it is a plasmid, is that it is self-replicating. It has its own promoters. It's ready to go as soon as it gets into the cell. So that plasmid, because it is what's bringing this gene of interest into the cell, is a vector. We didn't call it a vector in chapter 8 because in chapter 8, right, it's all in vivo. What just happens, right? Pieces of DNA go into the cell. But now we're switching gears and we're thinking from a lab tech perspective. And so we're going to call the plasmid a vector because it's not just going into the cell, it's going into the cell with the piece of DNA that I want to go into the cell. So the vector is the plasmid. And most of what we're going to be talking about in this chapter, in this class, is plasmids as a vector, because if we're using plasmids, we don't need to worry about transposons, we don't need to worry about Rep-A. We know that when the DNA gets into the cell, it gets in functionally. So now we have, right, what we would have already called a recombinant cell, right? Donor DNA transferred nakedly, so that's transformation, and now we have a recombinant cell because there's a recombinant piece of DNA inside it. And the cell, because that's a piece of DNA, will replicate it, no problem, right? E. coli knows how to replicate DNA. E. coli will also transcribe and translate that gene into the protein, right? So regardless of what you're going to do with the protein, this whole process, and that's why I cut off the bottom of it here, this whole process is the same. And this is a very commonly used process to get DNA into a microbial cell so that that cell can make some gene of interest into a protein of interest, right? For many, 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 many different reasons, right? That's the overall process. So that's what we're going to spend some time talking about because there's a microorganism involved in that process. And because even this process here, right, when I said cut out the gene, right? So if I want to have a recipe, right, you ever go to the doctor's office and you're flipping through the magazines, there are always cookbooks, right? There are always recipes inside them, and if you're a bad person, you will tear the recipe out of that person's magazine, which sucks for the next person that comes to the doctor's office, and it's like, oh, that sounds, right, so you can take a picture, right? But if you're gonna tear it out, or cut it out, you'd need a pair of scissors, right? Well, how do you cut a recipe out of a DNA cookbook, right? Imagine it's a teeny little molecule. You can go in there and read it and snip it with scissors, right? That would be kind of hard. Well, luckily, there is an enzyme that will do just that, and we call them restriction endonucleases, or restriction enzymes. And they are super cool enzymes who actually grab onto a piece of DNA, move along that piece of DNA, and cut the sugar phosphate backbone of a piece of DNA, not just willy-nilly, but in one particular place. So here are a couple, actually more than a couple, but not quite several, right? Here are 4 examples of what are called restriction enzymes. I'm going to draw your attention to this guy right here. So this guy right here is called EcoR1, and it's a great name because it's very descriptive. It was the first restriction enzyme that was ever identified, ever discovered, and guess who makes it? E. coli. So we call it EcoR1. Now, these are also named for the organisms that they were isolated from, but we're going to focus on EcoR1 because it's a very commonly used enzyme, and it's also one that you're going to be using in lab in a week or so. Now, EcoR1 will grab onto any piece of DNA, and it will hold on, and it will read the piece of DNA, right? Like you read letters, it reads all the C's and T's and G's and A's. And when it gets to this sequence of DNA right here, this word, it cuts the sugar phosphate backbone there, and it cuts the sugar phosphate backbone there, and what that does is make the DNA cut in half right there. All you have to do is cut the sugar phosphate backbone, and the helix will unwind, and the piece, if you're talking about a linear piece of DNA, the piece of DNA will be cut in 2. If you're talking about a circular piece of DNA, that will become linear. Now, why does that exist? Seems like kind of a weird thing to have, right? So E. coli is not going to waste its time, right? Because we know every time you make an enzyme, that takes bucks, right? It takes ATP. So why do they do this? They actually do this in order to protect themselves from viruses. So when a virus infects bacteria, the virus will insert its DNA into the bacterial cell, and the only defense that the bacteria has is to chew up that piece of DNA before it can cause any damage. So in vivo, in real life, what are these used for? They're kind of like a bacterial immune system. They're a defense system. They protect bacteria from infection by viruses. But when they were discovered in the 1970s, somebody said, that's a super cool thing that I can use to cut a piece of DNA at a particular section. And not just because I want to cut it up, but because I want to cut out that gene of interest. And if that gene of interest has a GAATTC on one side and a GAATTC on the other side, now you can cut it out, right? So these restriction enzymes are used like molecular scissors, right? And not just like a little kid that's going to go, right? They're going to cut along the lines and cut where they're supposed to cut. And they can be used to create recombinant DNA molecules because when they cut, right, what you end up with is a piece of DNA. I just want to make sure I'm writing it in the same order. And then you have another piece over here that is AATT, right? AATC. I just don't want to write it backwards and then have to be like, right? And a G. Yeah, there we go. So what you end up with is this piece of DNA and this piece of DNA, and what does that A want to stick to? U. If it's DNA, it wants to stick to a T, right? If it's RNA, it's a U. So this wants to stick to a T, this wants to stick to a T, that wants to stick to an A, and that wants to stick to an A, which makes it sticky. Because if it encounters a TTAA sequence, it will grab onto it. Same thing with this one over here. He wants to stick onto AATT, right? Which makes sense, because where did they come from? They came from complementarily bound pieces of DNA, right? So what you can do is you can create a piece of recombinant DNA by cutting the gene out, with that one restriction enzyme, creating these sticky ends. And then, right, so that's what you see up at the top. So this little purple guy, that's your gene of interest, right? That's the human insulin gene. Amongst all 3 billion nucleotides in the human genome, there he is, right? So I take that, and it's sticky, and it wants to attach to something, and I take a plasmid, and I cut a plasmid with the same restriction enzyme, but the plasmid only has one place where GAATTC is. So the plasmid goes from this to this, and it's sticky. So when it's sticky, and I put it into a test tube with this piece that's sticky, then they will stick to one another, and I can end up with a recombinant plasmid. So the one thing I'll say about this picture is it's not represented, right? It's not size represented, because the plasmid is going to be this big, and the piece of DNA is going to be this big, right? So this is the gene of interest. This is the plasmid, right? So you've taken the plasmid, opened it up, put it in this test tube with a piece of DNA, and because everybody has these sticky ends, they will stick. And you end up with a recombinant plasmid. So if we go back and we look at that picture, that's how, right? So we opened him up with one side, boop, and then we inserted right into those little sticky ends your piece of DNA, and now you have a plasmid that knows how to teach a cell how to make insulin. So now all you need to do, right, is that transformation process. Whoop, get it into a cell, it will start making the protein, and yay, you can start treating people not with pig insulin, but with human insulin made by bacteria or other prokaryotes, right? So those guys, can be used to make these plasmids, right? So how to get a plasmid that happens to only have one restriction site, one place where EcoR1 will recognize and cut it? Well, you make one. I mean, you don't make one, but somebody makes one, and you buy it from them. And the most simplistic of all of these, right, is a plasmid called PUC19. So PUC19, right, if we zoom in on the plasmid, we know that plasmids have to have an origin of replication, right? Super important. You don't want to go through all the work of making this plasmid and then putting it in a cell and having no way of making copies of the plasmid, right? We also know that we want that plasmid, right, to have a promoter on it so that we can make the protein that we put into the plasmid, and that exists over here. But now we want to be a little more sophisticated, because if we're trying to grow E. coli that know how to make human insulin, let's say that 1 in 1,000 E. coli cells actually gets transformed. Do you want to house and feed the other 999 bacterial cells that aren't doing anything for you? You don't, right? You want to find some way of killing them and only keeping the one that's going to do the job. And you know what you do? You teach the one how to be resistant to an antibiotic. And now when you grow him in the presence of the antibiotic, and the 999 die, and he survives, and so you're only feeding and housing the bacteria that have learned how to make the gene that you want to make. So this kind of a plasmid, right, makes a great vector because it will replicate when it gets into the cell. It will express the gene that you've put into it, and you can select for only those bacteria that have been transformed, right? So we call these plasmids, right, they all are derived from a plasmid called PUC19, right? And you're actually going to use one of these plasmids in a few weeks. On the 1st of October, you are going to be doing a transformation with a plasmid called pGlow. And the pGlow plasmid contains something called the BLA gene, which sounds kind of weird, but it codes for beta-lactamase resistance, which is resistance to penicillin or ampicillin or amoxicillin or some other beta-lactam antibiotic. So we can grow them in the presence of an antibiotic, and we can only allow those cells that have been transformed to grow. But what do we have on here that allows us to make this a recombinant plasmid? Well, notice over here, within an operon, by the way, you have a Hindi 3 site a BAM-H1 site, and an EcoR1 site, which means you have, oops, wrong way. This sequence right there, this sequence right there, and this sequence right there. So you can take that piece of DNA, and if you expose it to EcoR1, it will open up the plasmid and do nothing else to it, and it will make those sticky ends. And then all you have to do is add your gene of interest, and now you have a recombinant plasmid, and it just so happens that it is recombinant right where that cell will turn on transcription of the gene. And you will get transcription of the insulin gene and translation of the insulin gene, and you will get insulin produced by the cell that can be extracted from the cell, right? So in your scheme, which you're going to be doing in lab 14, you're going to be doing a transformation. And we're not just going to see what happens, we're actually going to try to make that transformation more likely than it would be in vivo. And we can do that by beginning with cells who really, really, really want to bind DNA from the outside. We call those cells competent, right? So not just any old E. coli, but E. coli that have tons of receptors for DNA. And then we're going to incubate them with the plasmid, right? Because it's a transformation, it's just plasmid. And then we do this funky thing with heat where we make it really hot, really cold, really hot, really cold. And that helps the DNA to get inside the cell. And then when we glow them, because we grew them in the presence of ampicillin, they will grow in the, we grew them with this plasmid. We put them in the presence of ampicillin, so everybody else dies. We have that promoter, so now we will transcribe and translate our gene, and you will change the phenotype of E. coli to make them fluorescent under UV light, which is not human insulin, but that's exactly right. I mean, insulin wouldn't be really helpful because you can't see insulin, right? You would have no way of identifying and going, ooh, wow, look at what I did. I made a cell that makes insulin, because that cell looks exactly like the cells that didn't. But you can pretend, right, every one of these, if this was the human insulin gene instead of the green fluorescent protein gene, each one of those would be insulin producing. And you grab it, and you would get some insulin from it. So this process of transformation is the simplest way for us to get a piece of DNA into a recipient cell, and it's easy because it involves some restriction enzyme manipulation of the DNA and then a transformation that has been done. So when you guys do this in lab, you are not going to be doing the restriction enzyme work. You're going to be using a plasmid in which a gene of interest has already been inserted, right? Because this isn't a biotech class, it's a microbiology class. We don't want to spend 4 weeks making a plasmid and getting a plasmid into cells. So instead, we are going to use a plasmid that's already been engineered. It already has a gene of interest, and your job is just going to be to get it into the bacteria and see if you can do that successfully. As an example of transformation. Yes?
The HIM3 and BAM, if those are used, are those also?
So you could use that. So let's say that there happened to be an EcoR1 site right in the middle of your gene. So if you tried to use that, it would actually cut your human insulin gene in half. So you would, oh, I can't do that. So you would try a different restriction enzyme. So that's, and actually in these multiple cloning sites is what they're called, right? Multiple cloning sites, because that's the point at which you can clone the gene. There are typically like 9 or 10 restriction enzyme recognition sites there, so that you as a researcher can say, this is what I cut my gene of interest out with, so I'm going to use that restriction enzyme to cut the plasmid. All right, so that's transformation. So what is one vector that can be used? A plasmid. Another kind of vector that can be used is a virus, which sounds funky, right? We're talking about using a virus, which only ever always is a pathogen, to deliver DNA into human cells, right? Which if I say I'm going to infect you with a virus to make you better, you're like, I don't think so, because that's not how viruses work. Viruses don't make me better, viruses make me worse, right? So here's the thing, right? Remember that a virus, when we looked in chapter 8 at transduction, that virus could accidentally pick up bacterial DNA and inject it into another bacterial cell, right? It's not like it knows what's inside. It's just a syringe. It doesn't know what it's been loaded with. So you can actually use this therapeutically to cure genetic diseases. It has a very narrow spectrum of use because you have to have a way of fixing all of the cells in a system in order to make a genetic disease go away. So one where this has been successful is a disease called severe combined immunodeficiency syndrome. And severe combined immunodeficiency syndrome, thankfully, though for some reason, right, one single recessive issue in one gene kerbolixes the entire immune system of a person, and if you don't live in a sterile environment, you will catch an infection and you will die. So people who are born with severe combined immunodeficiency syndrome have a very short lifespan, even when you try to keep them. It's sort of similar to an area where, if you've ever known someone or heard about somebody who's undergone radiation because of leukemia or something like that, and they have to be kept in a sterile room while their immune system recovers, they have to live their whole lives like that. But thankfully, someone realized that that was because of one bad gene. And they were like, I wonder if I could replace that one bad gene with the right gene, and now that person's immune system would function normally. So they started looking into this, right? And the reason that this doesn't have far-reaching implications for all genetic disorders, but it works here, is because severe combined immunodeficiency syndrome causes damage in cells in the bone marrow. Right? Not in your pancreas, not in your lungs, not in your liver, not in your kidneys, but in your bone marrow, where you happen to have stem cells all the time for your whole life, which we'll talk about more in chapter 16 and chapter 17. So you can take these baby stem cells from a person's bone marrow, fix them, and put them back into the bone marrow, and for the rest of their lives, though they're still making bad cells, they're also making good cells. And because it's a recessive disorder, the good cells can mask the bad cells. So in this instance, right, so you take now the gene of interest is this severe combined immunodeficiency gene. You cut it out of a healthy person. You use restriction enzymes to do that. You insert it into a virus in which you have scraped out all of the viral DNA. No viral DNA in there, only this correct gene. And then you do this very strange thing, right? Because this is a person who has no immune system. You take some of their bone marrow out. And thankfully today, right, you don't even have to take bone marrow from bone marrow. You can take shots and it will release bone marrow cells out into your bloodstream, right? But regardless, you take those bone marrow stem cells out and you infect them with the virus. But remember, it's not infecting them with viral DNA. It's infecting them with the correct human DNA, and then you just put those stem cells back into their body. And when you do that, those stem cells grow into more cells, which grow into B cells and T cells and natural killer cells and macrophages and phagocytes and all of the other cells of the immune system, and they all know how to work because they learned how to work. And they will mask the presence of all of the bad cells that don't know how to do their job. And so that person will live a almost normal lifespan, right? They tend to have a higher rate of cancers than other, people who don't have severe combined immunodeficiency, but we're talking about people who have a higher rate of cancer but live normal lives, like as kids play on playgrounds and eat dirt and do all the things that kids do, right? Rather than kids who live, right, if you're old enough, you might remember John Travolta played The Boy in the Bubble. It was based on a true story of a young teenager who lived in a bubble, right, basically a sterile environment, and died anyway. So more risk of cancer, not a perfect option, but using a virus to move DNA into the human cell, right? So the reason we're talking about this is not because of the disease, because it's a genetic disease. The reason we're talking about this is because a virus has been used as a vector to move the correct gene into the person who only has the faulty copy of the gene. So plasmids can be used as vectors, and viruses can be used as vectors. All right, so now some applications based on things that bacteria produce, right? And one of them is a really cool enzyme that was discovered in, well, you got the Nobel Prize in the 90s. I don't know when it was discovered. But an enzyme called Taq polymerase, right? Any idea what a polymerase is? It's an enzyme that makes copies of a nucleic acid, right? So Taq polymerase is actually an enzyme that was isolated from Thermus aquaticus, which is why it's called Taq polymerase. So Thermus aquaticus, what kind of environment do you think he might live in with the genus name Thermus? Hot. Very good, right? So he actually lives in an environment and his optimal growth temperature is about 75 degrees. So where your enzymes would denature, where E. coli's enzymes would denature, where all of the mesophilic enzymes would denature, he's like, sweet, I love it. And he can withstand temperatures all the way up to boiling. And the discovery of this enzyme from a bacterial cell revolutionized in vitro copying of pieces of DNA. Because if you think about it, right, so E. coli is sort of the lab rat of the world, right? So E. coli has been known for a long time. It's been used since the 1970s. But if we took the polymerase from E. coli, we wouldn't be able to use it for very long in a lab because it can only operate at 37 degrees Celsius, right? Which is not super, I mean, it seems like it'd be helpful, but it's not, because in order to in vitro copy pieces of DNA, you have to mechanically separate the strands of DNA, right? In vivo, DNA helicase would do that, which you're like, what, right? That's okay, that was general biology stuff, but you wonder, how do we get to the bases, right? DNA helicase opens it up. That's in vivo. How do we get to the bases in vitro? We have to cook DNA. And we have to make it hot in order for DNA and enough thermal energy for the molecule to open up and expose the bases. That takes about 100 degrees. So what happens to your enzymes at 100 degrees? And E. coli's enzymes? They're gone. But Thermus aquaticus is like, nah, I mean, that's not optimal for me. I'm not going to work at that temperature, but I can withstand that temperature. So you can do this procedure called PCR, or polymerase chain reaction, where all you have to do is put all the ingredients into a cute little teeny little tube, which you're going to get to do in a week or so. And you put it in a machine and you hit start and a chain reaction occurs. And at the end of that chain reaction, you have bazillions of copies of DNA. And the reason that that works, and the reason that Kary Mullis got the Nobel Prize for it, is because of Thermus aquaticus. Thermus aquaticus can withstand the experimental parameters of PCR. PCR operates with the experimental parameters of a high of 94 degrees and an absolute low of about 50 degrees. But it optimizes at 72 degrees. And that fits Thermus aquaticus perfectly. Thermus aquaticus says, got it, no problem, right? So what does PCR look like, right? So PCR begins with a piece of DNA that you want to copy, and then you open that piece of DNA up by cooking it for a long time, right? So it opens it up, doesn't break it, right? Those strands are still together, the sugar phosphate backbone is still together, but it denatures, they call the DNA. But we have to remember, denaturing DNA is not like denaturing proteins. Denaturing just means making it single-stranded. So here you have your two pieces of DNA, and you have your Thermus aquaticus DNA polymerase. But how does DNA polymerase know where to start copying DNA? Remember in chapter 8 I said it needs a primer. It needs that OH group, which it's not going to have if it's a single-stranded piece of DNA. So you have to add primers. And when you add those primers, they are tiny, single-stranded pieces of DNA who are complementary to your template strand, right? So here's your target, and these little yellow things are your primers. Notice the primer is complementary to this piece up here and complementary to this piece down here. Now you might have thought that was a bummer, right? 'Cause you're like, oh, now I gotta add something else to the equation. But what that means is you can pick the exact spot from the entire chromosome that you want to copy. And that is true of a 3 billion nucleotide genome of a person or a 3 million nucleotide genome of a bacterial cell. You can choose amongst all of those 3,000 genes and only copy one gene from them because you've made a primer, right? And these are really cool. You can, or I mean, you don't even have to make them. You just call the company and you say, I want a primer that is AATTCCGG, you know, blah, blah, blah. And they plug that into a computer, hit start, and they have a machine that actually synthesizes pieces of DNA. Super cool. So you do that, right? So you allow those primers to attach. Now DNA polymerase has a target, right? So now DNA polymerase can come over, it can copy the piece of DNA, and now you have 2 copies of DNA. And so you can ramp the temperature up again, denature the DNA, allow the primers to attach or anneal, and then allow them to be replicated. And that process happens over and over and over again in a machine called a thermocycler, which cycles through those temperatures. So it cooks it to denature it, right? So step 1 is to denature the DNA at about 94, 95 degrees. Then you lower the temperature down to somewhere between 50 to 65 degrees to allow those primers to anneal. And that's the second step, annealing. And then you raise the temperature to 72 degrees because that activates DNA polymerase. And DNA polymerase will then copy the DNA in what's called an elongation step. So this process, right, denatures, anneals, and elongates, right, or copies the target DNA. And at the end of one cycle, for every one piece of DNA, there are now 2, which means exponential growth. And in the next cycle, 2 become 4, and 4 become 8, and 8 becomes 16, and it doubles and doubles and doubles and doubles and doubles. And if bacteria can become over 1 million cells in 20 hours, or 20 generations, which takes 7 hours, this takes a few minutes. So one piece of DNA can become over 1 million in an hour. And why is that important? Because one piece of DNA is tiny, right? You can't see one piece of DNA, but you can see millions of pieces of DNA. Do you ever do, I think they still do this in general biology, you could like chop up onions and you add like alcohol and you add detergent and you break the cell and you add protease and you add alcohol and then you start spinning a glass rod and this white stuff will come out of solution. That's DNA. It's just millions of copies of DNA. So if you have millions of copies of DNA, it's actually visible. This process allows you to have millions of copies of DNA. Now that has far-reaching implications, but what do we care about in general microbiology? Number 1, it's a microbe. So we go, yay, it's another place where microorganisms have been found to be useful, not just in microbiology, but everywhere. The other thing about this process, bless you, that we talk about in general microbiology is because we can use this process to detect pathogens. Not because we can grow them in a lab, right? Because like a virus has to grow inside cells. So can you grow a virus on a petri dish? Bless you. You can't grow a virus on a petri dish, right? You can't streak them out and grow them because they need a cell to grow inside. So how do you diagnose a viral illness? A lot of times what you do is they will swab the person, right? If, let's say you have a cold sore and they're trying to decide whether it's herpes. They swab it and they send that swab off and they try to find herpes DNA in the swab. If there's herpes DNA in the swab, guess what's making your cold sore? Herpes, right? Same thing when you get a pap smear, right? A lot of times those are DNA tests that are done, right? They do the pap smear, right? Super fun. Grab off a sample from your cervix and look for bacterial DNA that would indicate gonorrhea or chlamydia, right, which are sometimes asymptomatic sexually transmitted diseases, right, which can cause big problems. So we can look for pathogen DNA in a host because we're using primers not for human cells, but primers for pathogens, primers for viruses, primers for bacteria, right? And we do that, right? And you're going to be doing this in class. You are going to PCR 4 different samples. And in that respect, we are going to be trying to solve a food poisoning epidemic, right? We're going to have a sick patient. We want to know who made him sick. So we can go shut that place down, right? So you are going to take these samples, right? And you are going to make billions of copies of DNA from the patient's sample and from these 3 restaurants. And now you need to look and compare that DNA, and you can't compare them in test tubes, you compare them using something called agarose gel electrophoresis, which I know you guys do in general biology, right? Where you have this gel, it kind of looks like clear jello, and you separate things on that gel into distinct bands. I think in general biology they do that with dyes, but in this class you're going to get to do it with DNA. So ours, right, we have these cute little gel boxes that you're going to get to use, and you're going to put all of your PCR samples into that box, and then you're going to apply an electric current because DNA has a negative charge. So when you apply that electric current, you're going to put a negative charge right here and a positive charge right there. And so the negative charge at the top will repel the DNA, and the positive charge at the bottom will draw the DNA toward it, but it has to move through this agarose, so big pieces take longer than little pieces, and let's see. Right, can I get it to go? That's a time lapse of what you're going to be doing. Notice pieces of DNA are moving, and they're moving according to their size. And notice as they move, they start to separate out into distinct bands. Each of those bands has a particular size of DNA, longer ones at the top, shorter ones at the bottom, and that creates a DNA fingerprint. Just like if you were to take your finger and put it in ink and then put it on a piece of paper, the swirls and whirls and whatever else all the other things are in your fingers, right, that make your fingerprint. Just like nobody has your fingerprint, nobody has the same DNA fingerprint as you either, unless you have an identical twin, who incidentally will not have the same fingerprints as you, but will have the same DNA fingerprint as you. No cell, no pathogen has the same DNA fingerprint as another pathogen, so we can look for a particular DNA fingerprint, like herpes virus, and say, if that fingerprint is there, that means that organism or that virus is there, which means that's what's making you sick. Much like, right, if you have an unknown fingerprint on like the murder weapon, and then you fingerprint the suspect, and that's his fingerprint, you go, you had that in your hand, most likely you're the one that stabbed somebody with it, right? So we can use DNA fingerprints to match an unknown sample to a known. So what can we do that? There we go, right? So how can we use that clinically? This is an example, right? So the coloring is a little bit different on this gel, but this is an example of pathogen detection using PCR. So in this instance, we have a bunch of patients who are all growing E. coli. So here's the problem with E. coli, right? E. coli can be your friendly neighborhood lab rat, right? We grow them all the time. You've grown him several times. He doesn't make you sick. He's part of the normal flora, right? That's where he comes from. But some strains of E. coli have additional genes which make them pathogenic. So notice each of these, right? So this EC at the end of each of these names is E. coli, right? So there's enteropathogenic E. coli, enterotoxigenic E. coli, enteroinvasive E. coli, and V is verocytotoxigenic E. coli. These are all bad news E. coli to get. They will make you sick. But the question, right, if a patient comes into the hospital and you swab their stool, which, yes, you have to do when you work in a hospital, right? You think that's bad, you're going to be the ones taking the stool sample, so enjoy. You streak that onto a plate and you go, oh, it's E. coli. Is that what's making him sick? We don't know. Is it normal flora E. coli, or is it one of these strains of E. coli? Well, you can look for a DNA fingerprint, because it turns out normal old E. coli has a very different fingerprint than all of these toxic forms of E. coli. So here is a negative control, because we always want to have negative controls, right? So there's no DNA there because that's a negative. This is the profile of this enteropathogenic. This is the profile of the toxigenic. This is the profile of invasive. And notice this is E. coli. So they have this in common because they're all E. coli. They have lots of other things, right? But this particularly they have in common. But E. coli lacks all of these pieces of DNA. now we have several patients and we want to know, is the E. coli that we found in your stool the thing that's making you sick? So patient number 1, we say, yippers, looks like it, right? Patient number 1 has a band that matches this. Patient number 2 and 3, E. coli is not what's making them sick because their E. coli profile matches normal flora E. coli, right? Patient 5, 6, and 7, right? So 6 has this profile here, 7 has this profile here, and then patient 4 ended up with a totally different profile, the DNA fingerprint of which matches that barocytotoxigenic E. coli. So we can use PCR when regular old microbiology doesn't work. When growing them up and identifying them doesn't give us enough information, we can look at their DNA, right? Because remember I said, I kept saying over and over again, right? Metabolism, growing them, testing them to see whether they can eat different things, that helps us to identify them because it tells us something about their DNA. If you can eat the food, you've got the enzyme, which means you have the gene, right? Well, all E. coli have those genes, but these guys have additional genes which don't have to do with metabolism, so we can't tell when we grow them. They have to do with pathogenicity, the ability to generate pathos or disease. So instead of looking for the effects, we look directly to the DNA. Do you have those genes? Yes, that's what's causing the bloody diarrhea, right? So that is one way that we can identify pathogens using PCR, right? So why do we care about PCR? Because it's a thermophilic bacterium that gave us the enzyme to do it, and because we can use it diagnostically to identify pathogens, which is something that we're going to see again in chapter 10 when we talk about identifying microorganisms, right? The last thing that we're going to talk about is what you're actually going to be doing with your PCR samples, right? You're going to amplify them, and then you're going to digest those amplified samples with restriction enzymes to make a kind of a DNA fingerprint. So you will take, right, from patient and 3 samples, so you have 4 DNA samples, 1 for each member of your team, and each of you to 1 sample will add EcoR1. And as soon as you add EcoR1 to it and put it at 37 degrees, because that's the optimal temperature for E. coli, EcoR1 will start moving down your piece of DNA looking for that GAATTC. And what's it going to do every time it sees it? It's going to cut it. So in this piece of DNA, there's a GAATTC here, and here, and here. And so cut, 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 which results in these 2 fragments. This one is shorter, so it ends up here. This one is longer, so it ends up here. But this one, right, so if this is the patient, and this is environment 1, when we try to digest this with EcoR1, there's no site there because there's a difference in the DNA there. And EcoR1 can tell the difference, right? Notice single nucleotide change. And E. coli goes, I don't know what that is. And ignores it and moves on by. And so you get a different DNA fingerprint. We call these kinds of fingerprints RFLPs, restriction fragment length polymorphisms. And you are going to be making restriction fragment length polymorphisms in lab during lab 16. Lab 15 will be you making the DNA. Lab 16 will be you cutting it and sending it out on an agarose gel to visualize the fingerprints and match the one environment to the patient, right? Figure out what made the patient sick. So you will do, that's one kind of a DNA fingerprint, right? PCR can be used by itself, like we saw with the E. coli, or PCR can be used in conjunction with restriction enzymes. And the reason that I chose to do the one with restriction enzymes for your lab is so that you can practice 2 of the things that we talk about in lecture, right? Because it's always nice to engage the information in lecture and lab before we have lecture and lab exams. All right, and that's it for chapter 9, right? So not everything in chapter 9. So really be careful because there's a lot of techniques in there that we don't talk about. Right? So if we didn't talk about it, ignore it. So now, right, so that was medical application. Chapter 28 is industrial applications. So one super cool kind of standalone concept in applied microbiology is a big concern, and it has been since time immemorial, and that is clean water. right? When people don't have access to clean water, people tend to die of terrible things because water is a vehicle for disease transmission. And one of the things that is in water that makes it unpotable, right, not drinkable, is microorganisms, right? So we actually use microorganisms to test for the cleanliness of water. And you're going to have an opportunity to do this in lab on when? On the 3rd, right? So at sort of the end of this unit, but before this unit test, you'll have the opportunity to do something called a most probable number test. Sound familiar? We talked about that in chapter 6 as a way of counting bacteria. This is its application. So pathogens can be transmitted in drinking and recreational water, right? We're going to have some fun talking about this in our human disease unit, right? Like cholera, giardia, all sorts of stuff gets transmitted through what's called the oral fecal route of transmission. But as you can imagine, it's not very common for a person's feces to actually come into contact with your mouth, right? Because that you'd see it coming and you'd be like, no, right? What happens is fecal material gets into water and it breaks up and becomes invisible, and microorganisms from that fecal material persist in the water, and then you drink the water. And when you drink the water, you ingest the microorganisms and you become colonized and you get sick. So an MPN test is used as what it's called a screening or a presumptive test to test for water or food cleanliness, right? To make sure there are not a lot of microorganisms in that water. And it starts with a water sample or a food sample. If it's food, you just dissolve it in water, right, so that you can get the bacteria out of the food. And as you might remember from chapter 6, you inoculate tenfold dilutions and you look for them to grow, right? So you inoculate the media, you look for growth. In this example, you get a 320, which means that your most probable number is 93 plus, right? But then you're like 18 to 420. Right? Because remember, that was one of the drawbacks of the MPN, was that there's this really wide range of values. So what you can do is something a little more sophisticated than an MPN. You can do a test called a CholAlert test, and CholAlert tests are really cool because they take this really wide range and they narrow it and they do something else. This tells you organisms. Now let's say that that's a lake or the ocean or a stream or a river. Do you expect that body of water to be sterile? No, right? Is it necessarily bad that there are microorganisms in that body of water? No, in fact, that's an indicator of healthy water, right? That there are organisms in the, I know that sounds gross. I mean, really, I'm drinking, yes, you are, all the time. So what you want to know is not just that there are organisms in the water, are there pathogens in the water, right? Is there fecal material in the water? That's your real question, because that's what's likely to make you sick, is organisms that made someone else sick, came out of their gastrointestinal tract and now are looking for a new home. So we can do something called an MPN test, a CholAlert test, because the MPN will only tell you organisms are growing. Now you want to look particularly not just for any old organism, but an organism that indicates fecal contamination. So it just so happens that coliform bacteria, who get their name because they were isolated from the colon, tend to be a great indicator of fecal contamination, because if there are coliforms in the water, there could also be fecal coliforms in the water. So if you see coliforms, right, you want to settle down, you don't want to be like, oh, blah, right? No, not yet, because there are also coliforms that live in the water. Now, should there be coliforms in the pool or in your tap water? No, because those are supposed to be held to a higher, right? You don't want to swim around in your pool, right? So we need to know not only are there coliforms, but are there fecal coliforms? And we have a really cool indicator organism who always tells us when there's been poop in that water in the last week, and that organism is E. coli. E. coli is an indicator of fecal pollution because he cannot survive outside the body for more than a week. So if there is E. coli in the water, regardless of whether it's the ocean, a stream, a lake, a river, your swimming pool, your drinking water, anywhere, there has been poop in that water within the last week. Do you want to swim or drink that water? Negative. Now, is it because you don't want to ingest E. coli? Probably not, because it's probably normal flora. But if E. coli is there, who knows what else is there? Something else that came from their colon that could be pathogenic. So E. coli is not looked for because he's going to make you sick. He's an indicator of fecal contamination, and if there's fecal contamination, there may also be other things in there that'll make you sick. So we just play it safe and we say if there's been poop in the water, we're not going to drink it and we're not going to swim in it. Right? That seems like a valid line to draw in the sand, right? I don't want to be anywhere where there's been poop, right? So you take this really cool test, right? You buy it, and it has two things inside it, right? It has food, so bacteria will grow. So you take a water sample and you add this powder to it, and you shake it up, and in that powder are two very important things, two pieces of food. One is a chemical called ONPG, and the other is a chemical called MUB. The reason that these two chemicals are really important as indicator is because E. coli, only E. coli has this one. Only E. coli has this enzyme which is capable of breaking down MUB. All coliforms have enzymes, right? Beta-galactosidase, which can break down ONPG. So when you add ONPG to your sample, if there are coliforms present, they will take that ONPG, they will digest it with their beta-galactosidase, and your sample of water will turn yellow. If that is the ocean, that's not a big deal. If that is your pool, that is a big deal. Recreational water can have coliforms in it, right? Pools shouldn't, because pools are chlorinated, right? So the question is, right, is the presence of coliform, right? So here, right, your little Venn diagram, here's all the coliforms, here's the E. coli. So are you out here or are you in here? So now you use this chemical, right? So this one tells you there are coliforms present when it turns yellow. If E. coli is present, his enzyme, beta-glucuronidase, right? Notice these are different. I'm not going to ask you what either of them is, right? But that's them. This will convert MUB to a fluorescent compound. So if your water, regardless of where it came from, fluoresces, you should stay out of that water and you should not put that water in your mouth, because that means E. coli is there. And if E. coli is there, that is an indicator of fecal contamination, and there could be all sorts of other things that are also there which are harder to grow and harder to see. So we use the presence of E. coli as an indicator of fecal contamination, right? So that's one application, industrial application of microbiology, is that enzymes, right, from microorganisms can be used to test for the presence of water contamination. The other application is really just the application of Chapter 7, right? Knowing what we know about what bacteria require for growth, how can we deviate from that and kill them, right? So in Chapter 7, we talked about physical methods of control, right? Heat, ultraviolet light, salt, pH. These are all things, right? All organisms, right? If you're a nutriophile and somebody makes it too acidic or too alkaline, you will die. If you're a thermophile and somebody makes it too cold, you can't grow, too hot, you'll lyse, right? If you're a mesophile, too hot, you'll lyse, right? If you are anything other than a halophile, if we put salt or sugar in something, you can't grow, right? We know that. This is the application of that. And these things have been happening for thousands of years. People have been noticing, right, that if they don't know why, but if they salt meat or dry meat, that it persists, right? It lasts. Cold has only been used since electricity, right? Without electricity, I mean, you had ice, right? But outside of the wintertime, there was no way to refrigerate, right? So drying historically is a method of controlling microbial growth on food, right, to preserve food. Osmotic pressure, salting things, sugaring things. Fermentation, right, allowing microorganisms to grow, like putting them in intentionally and causing them to ferment, which produces acids, preserves your food, right? And people have been tweaking this for a long, long time and adding particular things, and right, even today, people who make kombucha, right, they talk about having a mother, right, their yeast, that they like the way the kombucha tastes, so they save that yeast, right? Or before more sophisticated things like that, right, women who would make bread would take a piece of the risen dough before they bake it and you hold on to it. And you add that to the next batch of bread so that you always continue this beneficial fermentation, right? Today, of course, we are far more sophisticated. We have high heat, we have ultraviolet light, we have x-rays, right? Canning is one of the oldest, newer forms of food preservation, right? Been around for a long time. And it involves sterilizing food, aseptically transferring it to sterile containers. And right, if you have canned goods, those things can sit on your countertop or in your cupboard for years without spoiling, right? Same thing with glass jars, which have been sealed, right? So canning, which typically uses heat, just radiation can be used, right? You can radiate things with x-rays. And then if you put them into a sterile container, those things can be kept for long periods of time, right? Spices are often irradiated because if we cook them, we use heat to sterilize them, it changes the flavor of the spice, right? High energy electrons, high pressure, like these are all things, right? So we take chapter 7 and we apply it. Chapter 7 is just the understanding. Chapter 28 is the application of chapter 7. These are the ways that we can control microbial growth. How can we use that understanding to keep our food safe, right? So food preservation, right? Then, right, we have, again, like bazillions of different ways that we can use microorganisms to make things for us, right? So we can go back to that picture from chapter 9, right? And this is where chapter 9 and 28 really start to come together, right? Where we're talking about particular applications of the recombinant DNA technology that we talked about in chapter 9, right? So we can talk about that in food production, right? So microbes are used to make food, delicious foods, by the way. We used to have a thing, right? We were for a while trying to figure out if we could make beer and like make sauerkraut in class and then have it, right? But we can't. But cheese, right? The only way you get cheese is because microorganisms are rotting your milk. But they rot it in a delicious way, right? So you have lactic acid producers. Whenever you have a tangy cheese like cheddar, that's bacteria, right? They're taking the milk, the lactose from the milk, and they're converting it to acids, which give it tang, right? If you like blue cheese, that's molds, right? And some of the most delicious blue cheese on the planet is like in these caves in Spain, and they just take the milk and they stick it in the cave, and they come back a little while later and it has these beautiful ribbons of purple and green and blue. That's mold. It's delicious, and it comes from microorganisms, right? And you can't make any cheese at all without an enzyme that curdles milk. And historically, that enzyme came from the stomach lining of baby animals, right? Because they produce this enzyme naturally. So calves or baby sheep, lambs, they're called lambs, right? Kids, right? So like my kids and I read the Little House on the Prairie books, right? Super awesome. Have tons of microbiological applications because they get malaria. Her sister gets scarlet fever. And also because when Ma, right, when they butcher a cow, Ma gets super excited because they butcher one of the calves and she gets a piece of his stomach. And she takes that piece of his stomach and puts it into milk. And that starts the process of making cheese. And if you're horrified by that, don't worry, because guess what? You get that enzyme today. We taught bacteria how to make it. So you can, because you're like, I'm a vegetarian and I thought I could eat cheese, but you're telling me there's a cow enzyme in my cheese. No, there's not. They took that gene of interest from the cow, They inserted it into a plasmid, they put that plasmid into bacteria, taught them how to make the enzyme, and now you can use that enzyme, right? So renin, rennet, that one is the natural form, one is the made form. So you can't get cheese without microorganisms, right? Can't get bread without microorganisms because it's yeast, right? Have you ever watched yeast, right? When you put yeast in warm water with sugar, it makes this big frothy mess, right? And you can smell it. It's that smell that you smell when you go to pizza places, right? If you don't make bread. It's fermenting, right? It's fermenting that sugar to alcohol and carbon dioxide. So when you put it in with the flour, it starts to rise. And you're like, oh no, bread is alcoholic. Don't worry, because when you put it in the oven, the alcohol all goes away, but the bread bakes around all of those CO2 bubbles, and that's where you get the air in your bread. Is all of that trapped CO2, right? Incidentally, those yeast also are used for the alcohol portion of that fermentation process, right? In making wine or beer, right? The reason that beer is carbonated is because you seal those containers and you trap both the alcohol and the carbon dioxide, so you get a carbonated beer. The reason beer has a top alcoholic concentration is because alcohol is a poison. I know that hurts, but that's just the reality of it, right? So as they produce alcohol, they start killing themselves, and at some point, right, and you think, well, how do they have such a high concentration? Because they've been messing with yeast and training them to withstand higher alcohol concentration. So now you can get a beer and a wine that has a much higher alcohol concentration. Of course, wine, right, is not typically bubbly because it's open fermented, so the gas all goes away, and what's left is the alcohol, right? Unless you're talking about a sparkling wine, like a champagne or something like that, right? Those are close. And then, of course, right, I put this guy on here. You might be wondering, rum. It's not because it's my favorite type of, but it's thought to be one of the very first distilled alcohols. And this particular rum, my husband loves reading about weird, obscure things, and he got on this kick about rum. And this is the British Navy used to give an aliquot of rum to everyone on board ships for years and years and years and years. You can imagine it gets a little sketchy, right? So much more recently than you would think, they decided, you know, maybe we shouldn't do that anymore. And they sold the recipe to this company here, and so this rum is the rum that is actually the recipe that was a proprietary secret recipe for like 100 years from the British Navy. So, la-di-da. So lots, and you can, when you just look at that and you go, that looks like a party, right? Right there. Got some wine, got some cheese, got some bread. I mean, what more could you ask for, right? A designated driver. All right, so these are ancient things that have been done, right? Beer is ancient, really ancient, right? I think Egypt, they made beer. It was more like porridge back then, but they made beer. What about more recent advances, right? So I was doing some fun stuff this week, and if you downloaded your notes a long time ago, you're like, where did that come from? Because I can never stop. My daughter says, do you ever stop changing things negative? Right? Because you got to keep it fresh. You can't do something for 20 years and just keep, I mean, you could, but we've all been in those classes, right? With the person who just has the same slide that they've had for 20 years. So I was fiddling around looking at some stuff. So here are just a few of the industrial proteases, right? So protein enzymes that have been discovered and are used for various industrial applications, right? Some of these are horrifying, but they are what they are, right? So they've actually been looking at proteases to digest biofilms. Remember, we talked about biofilms, right? How do we get rid of biofilm? Not in people, unfortunately, but in industrial settings, you grind them out mechanically, which may prove impossible, in which case you have to take it and throw it away. But they've been looking at biofilm remediation, using microbial proteases to chew up the biofilm, right? Silk degumming sounds nasty, right? But have you ever, silk, right, is the cocoon of a silkworm. Remember we talked about that, right? Because this was a big push in early epidemiology. It was not human disease, it was silkworm disease, right? Pasteur was very interested in silkworm disease. Well, you can imagine a cocoon, right? Well, where do you get those beautiful silk fibers? you have to digest all of the nasty stuff around them, and that's called degumming, which right now is an industrial process that takes lots of chemicals, but you can use a microbial enzyme to digest all of that material and just be left with very nice silky strands, right?